So, Erin, tell us about your collecting technique.
My collecting technique or my collecting?
Your collecting and your technique.
Well, yes, colleagues.
Well, step one, find a lichen.
We're in the rainforest here after Micrandra camp, towards third camp. A wet rainforest full of bryophytes, cyclanthaceae, Micrandra, rubiaceae, looking for lichens, which are sparse in this kind of environment. Although bryophytes are very rich here because it's very wet. So here we are investigating some splot on a tree to see if it's a lichen of interest. Is it reproductive?
It is, but we just collected it.
Not new. Alright, I'm going to go on to the next tree.
And have you brought your trowel with you?
I did not bring my trowel with me. Alright, we're hiking up freshly cut line in virgin rainforest. Investigating a next tree with a little splot.
Alright, we can collect this.
Aha. Looks novel.
So,
Let's check this out.
whereas there has been botanical exploration of the Tepui highlands since the early 1800s, there are virtually no collections of lichens or bryophytes from the region. So, while we're here on this plant collecting trip, we are making an effort to sample the cryptogam, that is, lichen and bryophyte biota. So, we bring this wood chisel and a nice heavy hammer, which doubles not only for wood but also for hard rock specimens, and try to make some nice museum specimens. This would be perhaps half of a collection. Here we have to collect in high replicate numbers so we can distribute specimens to various museums.
The bark here is quite interesting in being rather red.
The bark
We
is
haven't
quite interesting. I was just
figured out
going
what
to-
sort of tree this is yet. Other than it's
Alex, will you have a
very,
look up
very
with your
tall.
binocs and tell us if this is a legume?
I would had I perchance brought the binoculars.
Oh, okay. Well, with bark this red and legumes as rich as they are, there's probably a 50% chance that that's what we're collecting on at the moment.
It
No
is also
sap,
surrounded by legume-looking seedlings.
such as what?
Oh, such as these little guys right here and right there. Most likely a papillion, says Alp.
So after collecting enough specimens for 5 or 6 museums, we'll just make note of the substrate and any other pertinent information. So this is on bark, obviously, and perhaps on a member of the bean family, Fabaceae. Onward.
I will sample the sapling here.
We got a 60-degree view of
So Alex,
the rainforest on the slopes of Kamakusa.
Yes, Erin.
tell us about what you're doing.
Well, we decided, sort of a little side project that we came up with while we were here, that we would investigate nodulation in some of the sessile taxa that are found here in the Guyana Highlands. Nodulation is known in some parts of the sessile subfamily. It's not a real subfamily, but so we're not really sure how many of them do it, and so while we were here we thought that we would sample some of these saplings and seedlings, which are easy to dig up, to see if we can find the presence of nodules. And nodules are they're sort of like a small home for nitrogen fixing bacteria that occur in some members of the legume family. Almost all papilionoids do form these nodules, most mimosoids, and as I said before, some of the sessile. So we're just opportunistically sampling these seedlings whenever we can find them and documenting whether or not we have found nodules. And then back in the States, we'll take a look at them and see if they are homologous to nodules formed in other parts of the family. And cut.
All right,
makes
let's
sense.
see the digging of this seedling.
Oh, sorry. Okay.
Now that we do not have a trowel, we're using a machete or a cutlass in the parlance of Guyana.
Yes, so the machete, or cutlass as Ken said, is a very useful tool to have here in the bush. Useful for digging latrines, cutting line, and sampling plants. Not in that particular order. So
the botanist when they get out of hand.
So we'll just
Cutting down little trees.
Sorry, little tree.
You smell sunflower.
Now, let's see. Oh, you son of a bitch. Oh, I shouldn't curse. Well, Ken, you should just back up. Safety first, second and third here in the jungle. So, sometimes you'll find that the soil is just so filled with roots as these plants are, fighting to get every last little bit of nutrition they can out of the soil that it can be quite difficult to dig down. This isn't like digging in your backyard where you find, several feet of loose soil. So let's see what we found here. So I'm just gonna try and peel this apart. Oh, and it looks like I found one, but it's detached, so I can't be sure. So let me just peel this apart. Now we probably won't be able to identify these down to species, but we should be able to get it down to genus just based on the leaf morphology.
We're also taking a DNA sample,
Yes.
which could be used for further DNA barcoding and further identification if this turns out to be useful for research.
And
Not much there, eh?
not much. Then
All right.
I'm
No nodules.
no, but it goes.
Let's check out this fern here, which is one we have not sampled before. This little creeping thing on the stem. Rip it off. It's climbing
in the
fern.
rainforest.
Pull it up. I have to peel off all the teridophytes, but let's go see what Erin has discovered over here. Let's traipse through. This is Cyclanthaceae here. Sort of looks like a palm, but isn't. There are various bromeliads. There's an philodendron, an Araceae. There's a anthurium there with heart-shaped leaves. And here we have some large lichen blotches growing on a tree. There's a melastome there, which is a vine climbing up. Don't quite know what the tree is yet. It's way, way up there.
We forgot our binoculars.
There's lots of this particular lichen. Oh, there it is, reproductive.
So these little orange pieces that you're seeing are the sexual reproductive organs of lichens, known as apothecia. Not all lichens reproduce sexually. Some reproduce asexually, like this one. These little green pieces are hyphae, fungal hyphae, and they break off and blow around in the wind and reproduce that way, land on a new substrate, germinate, make a new lichen.
This looks like the same tree as before with the dark red inner bark.
Chances are good.
Let's go around here while she chisels
somewhere.
away at its...
So, lichenology is a science like any other, but two of the most important features when collecting a lichen are referred to as the landscape factor and the sparkle index. And these particular lichens have a very low landscape factor. But
Landscape meaning relief that looks interesting.
we'll demonstrate landscape factor later. This might have nice landscape factor, this here leafy liverwort. But this lichen with these shiny orange apothecia has a very high sparkle index. Let me chisel a piece and see if, you know what, that's growing on something interesting.
Should I be taking a photo of this?
I'll take the next piece. How zoomed in does your video camera go?
Actually, that's pretty good.
Cool.
There you can see the little spots. Apothecia. Okay.
All right, good. So we'll collect 3 more specimens, 3 more species. This particular tree, and then move on.
Epiphytes up there, same philodendron, forming a big bird's nest. Let's do a 360 through the canopy here. Trees are all rather small, despite being virgin rainforest. There are no massive trees as you would think of in sort of large U.S. forests, virgin forests.
And why do you think that is, Dr. Wurdack?
Soils are generally poor here. Don't know what else might be a factor.
Soils in some parts of this Tepui range are the poorest in the world.
They're derived from quartz sandstones.
They've been around a very, very, very long time.
From the
So
Precambrian.
2 strikes against soil fertility, one being old age, so all of the nutrients that were in the parent rock have since weathered away and washed out to the ocean. Second being, we're in one of the wettest places on Earth, which equates to extreme weathering rates.
The soils here are very shallow. We're on a rocky sort of talus slope that's disguised by a rich humus and duff layer with lots of roots and rotten logs, but it's very thin. We've seen places where it's less than a foot thick before you hit solid rock. Lots of mosquitoes here at the moment.
Fresh rain.
Rained heavily last night.
How many inches do you think it rained?
It'd be nice to have a rain gauge.
6, 7 maybe?
We've had daily storms here at night for the past several days in this camp, which is in sort of a valley on the southwest side of Kamakusa, southwest side, more or less.
Picture of this camp before we...
All right, we're gonna pause for a minute so that we can take a SLR photo of this.